Hello students, welcome to EPG Pathshala. I am Vidya Kothekar and former professor from All India Institute of Medical Sciences. Also worked at JP Institute of Information Technology, Noida and D.Y. Patil Institute of Biotechnology and Bioinformatics in Pune. Today, we are going to talk on a module, Drug-DNA Interaction, a computational approach. It belongs to the paper on bioinformatics. After completing this module, you should be able to introduce yourself to basic concepts in drug interaction with DNA, enumerate quantum chemical studies on DNA binding hydrocarbons and interaction of metal ions with DNA. You should be able to discuss thermodynamics, molecular modeling, energy minimization, and molecular dynamics, MD, studies on drug DNA interaction. Describe and elaborate molecular docking studies on drug DNA interaction and also drug design. You should be able to do the most challenging task of predicting ab initio the docking site for a drug with DNA using a Monte Carlo simulation technique known as MC technique. Let me give you some basic concept in drug DNA interaction. Small molecules as drugs can interact with DNA by non-bonded, electrostatic and H-bonding interactions. For example, if you take metal ions, alkali and alkaline earth metal ions can interact with the backbone phosphate groups, while transition metal ions can form salt bridges and covalently bind to DNA bases. Small aromatic molecules as proflavin, acridine, and ethidium bromide can slide in between the base pairs and intercalate if their size is suitable. Heterocyclic aromatic molecules with cationic tails and protruding electron donors or acceptors can bind to DNA in minor and major groove by hydrogen bonding and electrostatic interaction. Whereas bifunctional antibiotics like economycin can bind in DNA grooves as well as intercalate. Minor groove binding drugs usually show preference to AT sequences. This is because of lower electrostatic potential and shape-wise suitability. How do molecules get groove preference? You can see here, major groove offers more specific contacts. We show here red oxygen acceptor, blue nitrogen acceptor, black NH donor, green CH donor. We show it in the major groove and minor groove for GC, AT, CG and TA sequence. So major groove, as you can see, offers more specific contacts for hydrogen bond formation with bases. However, because of the larger size of the major groove, these have less favorable van der Waals interaction. Presence of bulky amino NH2 group in the minor groove of GC sequences obstructs DNA binding in minor groove of GC sequence. The first quantum chemical studies on DNA binding were reported by Pullman's group in 1955. They tried to correlate carcinogenicity of polycyclic aromatic hydrocarbons, polyerenes, as benzpyrene and dibenz anthracene with electronic structure using a very simple method. valence bond approach, and Euclid pi electron molecular orbital method. The most fundamental quantum mechanical theory is KMN region. Later, they also proposed Bay region to explain transition between parents and epoxides. Quantum chemical studies using molecular orbital calculation method and supramolecular approach with EHT, CNDA2 and other were reported in our group by 1970s or like those years rather. We studied binding of sodium plus, Ca2 plus, magnesium plus with ATP and ADP. These were, I think, few of the first reported studies on metal DNA interaction. It was seen that these metal ions significantly influence the charge distribution pattern around phosphate group. The cleavage of the terminal phosphate in ATP was explained on the basis of these results. There have been many computations on metal binding to DNA bases. In our group, we performed conformation energy calculation CEC and molecular orbital calculations on binding of copper 2 plus with DNA bases. It was found that copper 2 plus modified charge distribution around nitrogen atoms of bases by charge transfer interaction. And destabilization of DNA by transition metal could be explained on its basis. Density function DFT, quantum mechanics QM, molecular mechanics MM. These studies were used by many authors. For example, QM-MM calculations were reported by Gaikon, not very recently, but in 2009 and in 2010. And they studied binding of platinum and ruthenium anti-cancer drugs with DNA. Calculations on cis platinum NH322 plus bound with CCTG GTCC duplex could lead to acceptable geometries, especially with explicit solvent molecules. Results explain carcinogenic activity of cisplatinum drugs. Thermodynamic studies on ligand DNA binding have been reported by a number of people. Chairs, Huck, Sheikh, actually, they belong to, one of them belonged to Dr. Jairam's group in India. Authors have analyzed drug binding in terms of free energy as an interplay between deformations in the molecule and entropy losses and favorable electrostatic, van der Waal and other contributions. These were calculated using theoretical methods as molecular dynamics. Molecular modeling and energy minimization studies. Molecular modeling studies on drug DNA interaction have been limited owing to the complexity of the system. Sequence-specific recognition for minor group binding ligands such as netropsin, distamycin A, H33258 dye, and lac repressor headpiece consisting of ALA, GLN, LU, ALA in beta sheet was studied in our group. Distamycin A with DA5, DT5, the backbone was aligned with the helix such that it bisected with the minor groove. The drug was allowed to slide along the helix axis and also had conformational flexibility. All the structures were energy minimized. Sequence-specific DNA recognition and influence of local excitation in DNA structure was analyzed in this distamycin study. Molecular model for interaction of proflavin in CpG site of DNA decamer GATA-CG-AT-CG was studied by Needle in 1988. The starting geometry was taken from crystallographic studies on CpG proflavin complex. Molecular mechanics with amber force field was used to obtain stereochemically acceptable structure. The structures were optimized using a method called EMPMDS. In the case of proflavin DNA interaction, it was found by Needle's group in 1988 that the grooves were wider compared to the normal B or A form of DNA. He calculated helix parameters using helix analysis program by Rosenberg and Dickerson. The authors systematically studied variation in local and global helical parameters. Several investigators used molecular dynamics method to design anti-cancer drugs. For example, Van Gunsten's group studied relative free energy for binding of two non-covalently binding ligands, nitropsin and distamycin, with CG-CG, AAAAA, CG-CG using thermodynamic integration method. Simulation for 2 nanoseconds was done. The results were found to reflect experimental studies. We show here schematic representation of hydrogen bonds between DNA and drugs nitropsin and distamycin. Nitropsin and distamycin with DNA was studied using Insight2 package. They started with two PDB structures with PDB ID 101D and 267D. This was used as a starting geometry. The complexes were solvated in periodic truncated octahedron water box, which was very different, with 11,034 SPC, simple point charge water molecules. DNA and ligand were positionally restrained using harmonic interaction with force field constant 2.5 10 to the power of 4 kilojoules mole per nanometer. Using pro-ion utility from Gromos, 20 chlorine minus and 43 Na plus ions were added. The same procedure was adopted for ligand with 3225 SPC water molecules, 7 chlorine ions and 6 Na plus ions. We show here nitropsin and distamycin in minor group, and we also show the root mean square deviation for nitropsin and distamycin atoms rather. You find both those drugs fit very well in the minor group of DNA, which is called snugly fitting drugs rather, and they are actually look as if they are designed for fitting into the minor group. The detailed analysis of structures and energetics led to a better understanding with the experiments. Relative free energy of binding between nitropsin and distamycin was minus 38 kilojoule per mole. It was little overestimated compared to the experimental value of minus 11 kilojoule per mole. Application of 3D database search to drug DNA interaction is more complicated. The docking tools ideally try to calculate activities of different chemical entities against a given target to yield high active ligand. The scoring can be done using simple scoring functions as H bonds, contributions by van der Waal and electrostatic interaction, or more complex force field as amber, chromos, char, CVFF, etc. Molecular docking technology was used by many authors who focused mainly on the protein-related system, restricted to rigid molecular approach for target, that is, proteins were considered as rigid, with very limited flexibility to the side chains of proteins in the binding area. Majority of these studies were biased against the pre-knowledge of binding site. That is, you have to already know where drug is supposed to bind. Limited conformational flexibility was introduced by some workers for the ligand. Otherwise, many times ligands were considered fairly rigid and only some angles were allowed to move. When used with the prior experiments, DOC, AutoDoc, and MOE provide computational filters and reduce the cost of experiment. So you can decide what experiment should be done or what drug should be taken for experimentation. Docking studies with drug DNA complexes are still limited. 5-fluorouracil or 5-FU is a fluoropyrimidine with marked anti-tumor effect and is very much used in the treatment of solid malignant tumors, particularly colorectal carcinoma. Its problem is it is toxic, and docking of some prodrug systems such as amines, alcohols, and peptides was exploited to improve the anti-cancer activity of this drug and minimize toxic side effects. 5-FU is known to interact with DNA by intercalation, but the exact mode of interaction of these other prodrug systems is not known. That's why this study was taken up. Cyclic voltmetry and molecular docking studies had been reported by a group of Queen very recently in 2014. The authors synthesized two types of structural similar molecules, 5-FuO and 5-FuM. The docking was performed using AutoDock 4.0 with Lamarckian genetic algorithm, LGA as it is called. Free energy of interaction was calculated using Amber force field FF03 package. For binding of 5-FU in ortho and meta analogs, the authors used the DNA sequence CG. The PDB ID of the structure was 2DYW with Gastier charges and added water molecule in a box size of 106 into 110 and 102. The binding was studied. They used the grid spacing 0.375 angstrom. A typical model for binding of orthoanalog is shown in this picture. 5-FuO and M analogs, the result shows that both ortho and meta, O and M analog, bind to DNA in minor bows only. They do not intercalate. On the basis of binding strength and free energy, it was found that the ortho form showed to be more suitable for binding to DNA. And these authors proposed that it can be tried as an anti-cancer agent. Docking of ellipticin and netropsin DNA was studied by Risi and Nets. They took 4 different models of DNA. First, crystallographic DNA without intercalation gap, crystallographic DNA with intercalation gap, canonical bDNA, and modified DNA with the intercalation gap. The docking was again achieved using AutoDock 4.0 with Lamarckian genetic algorithm LGA. The starting structural data needed for this calculation was taken, for example, ellipticin with the CG80CG from PDB-ID IZ3F, whereas with netropsin with CG8080CG was taken from PDB-ID 1DNE. The program could correctly identify two of the main binding modes only if the target possesses the artificial intercalation gap. Otherwise, it will only bind in by the groove binding. We show in this picture the best dot conformation of ellipticin and netropsin. The AT region in this figure is shown by magenta color and GC region in gray, and you can figure out where the two drugs bind. Docking of drugs to non-canonical DNA has been studied by a number of people. Ma et al. Performed virtual screening of ligands very recently in 2011 using programs as DOCK and AutoDock. They studied binding to DNA triplexes and G quadruplexes. Hypothetical model showing side and top view of the binding of a drug Fonsecin B16 with C-Miss G quadruplex is shown in this figure. Ma et al. Also studied series of metal complexes bearing molecular light switch dipyridophenazine DPPZ and their derivatives using number of biophysical and molecular studies, and it is just shown, some of them are shown in this picture. A typical example of how an antibiotics like actinomycin D or donomycin A bind to DNA triplexes is shown in the picture. Number of such compounds can stabilize DNA triplexes. Chairs in 2003 produced derivatives that displayed increased binding affinity towards antiparallel DNA triplex. Holtz proposed an alternative approach to identification of selective parallel DT with drugs. One of the most interesting study of ab initio docking of drug, because normally we expect a docking site and then start at that site, but with no previous information, was tried by Rowe et al. And in 2005, but they used Monte Carlo MC simulation method on reduced atom representation and all atom force fields and knowledge based potential. Interaction of a methylene blue with which has anti-malarial activity was studied with GC 80 80 80. The sequence has first a GC type of structure, then 80 type of structure. The reason was the methylene blue binding depends only on the base sequence and not the propensity to bind by intercalation in major groove or minor groove. We show in this picture snapshot of Monte Carlo simulation of methylene blue with DNA. Both drug and target DNA molecule were allowed to move relative to each other. Results showed 80 specificity in the minor groove, but the drug also intercalated with the alternate GC sequence. So, students, let me now summarize what we have learned in this module. We have described first some basic concepts in drug DNA interaction, introduced you to the quantum chemical studies on electronic aspect of DNA binding, DNA binding hydrocarbons like toxic carcinogens, and interaction of metal ions with DNA. We enumerated use of thermodynamic methods, molecular modeling MM, energy minimization EM, molecular dynamics MD, for drug DNA binding. We elaborated on molecular docking studies on drug DNA interaction. Lastly, we introduce you to the most challenging field of ab initio docking of drugs to DNA using Monte Carlo method. Thank you.